Okay, so we'll get started. Thank you. Okay, so I'm sometimes mistaken for coming from Australia, but our speaker today really is from Australia. So Dr. Jane Hill is an assistant professor at the University of Vermont. She obtained her bachelor's degree in chemical engineering and a master's degree in environmental management, both at RPI. And later, she did a PhD in chemical engineering at Yale University. She's going to talk to us today about small molecules that can tell big stories using volatiles to rapidly diagnose infectious agents. Please welcome Dr. Jane Hill.
All right. Thank you very much for having me. I appreciate being here and sharing some of the really cool work that we're doing in my group with small molecules. And so, as Pasteur is quoted as saying, the role of the infinitely small in nature is infinitely large. And that holds true today just as much as it did back then. And as I was introduced, my name is Jane, and I'm at the University of Vermont. I am in the School of Engineering at the University of Vermont, as well as affiliated with a Center of Biomedical Research Excellence in Immunology and Infectious Diseases, so I work a lot with the medical school there. And I thought I would take a moment and say that if you have questions during my talk, just raise your hand. If I don't see you, wave it around. I'm happy to answer questions along the way. Does anybody recognize this guy at all? Oh. That's okay. You're about to hear about him. This chap here is John Fenn. Now, does anybody know who it is at all? No, this is terrible. He's the first chemical engineer to win a Nobel Prize. Pretty cool. He earned his Nobel Prize in his 70s. He did his Nobel Prize winning work, in his 70s. So there's hope for all of us. Yeah, and the way that he did that is that he's the guy who did electrospray ionization mass spectrometry, which means that he was able to make molecular elephants, proteins, fly into the mass spectrometer. So he was able to charge them up and send them into a mass spectrometer, which is what we use today to do many things in terms of protein analysis. And the way that he did that is he would dissolve the protein in a slightly acidic solution, and then that protein would take on a charge through a proton transfer reaction, and then through evaporation and Coulombic fission, the protein would be charged, or fragments of it would be charged, and sent into the mass spectrometer where it could be monitored. On his way to doing his Nobel Prize winning work, Fenn actually found that he had some problems with his system. And what he found was that room air was getting into his electrospray ionization system and messing up his signals. So he sealed his system and then went on to do his Nobel Prize winning work. Well, it turns out that I sat down, had the privilege actually of sitting down with Dr. Fenn and some other engineers who were pondering electrospray ionization mass spectrometry and the fact that there were these contaminants from room air that could be measured using this technique potentially, this error that was irrelevant for the Nobel Prize. And the folks that I was talking to weren't biologists, and they wanted to consider using breath as a way to detect cancer. And by using the fact that you could measure these volatiles in the electrospray system. And I didn't know much about it at the time, did some mass spectrometry, but I said, well, what about the microbes that are in the oropharyngeal area? How are they going to influence these volatiles that you think you're going to measure from the lung when you're looking at different types of cancers rather? And therein started a path that I've been on now for the past few years, where I've been looking at the idea of using volatiles that are produced by microbes as a way to figure out who they are, how many they are, their metabolic status, and so on. And it turns out that physicians have been using breath as a way to diagnose all sorts of things that are going on in the human condition for a long, long time. This is the Hippocratic Oath. It's all Greek to me. Sorry, I can't resist that. And it turns out that Hippocrates was able to write about things like smelling the sweetness of breath, so acetone, which is associated with what condition? Does anybody know? Diabetes. You can smell nasty, fishy things associated with poor liver function and so on. And in some cases, you can relate it to infectious disease. Not that they knew about microbes directly at that point. Lavoisier also measured breath, although he was mostly concerned with respiration processes. As for me, I like to smell things, including flowers, but in particular, I really like to smell bacteria. I know that sounds weird, and you may not let it go out of this room, but bacteria sometimes have a very characteristic smell. And so the goal of my group, the grand goal, really, is infectious disease diagnostics, being able to figure out what somebody has, what's making them sick when it's an infection, using volatile analysis. In particular, we focused on the lung, ultimately for breath analysis, but initially maybe just to learn about one human-related infectious disease system. And it turns out that if you have a lung infection, that the way in which people figure out what it is that you have that's causing your disease is you cough up stuff. That stuff is called sputum. It looks like that. It is as vile as you might imagine. And then they take that sputum, they break it apart a little bit, and then they spread it onto a plate. They grow up colonies on that plate. They transfer those colonies onto another plate that's a selective media. Maybe it's selective for Staph aureus, maybe it's selective for Pseudomonas. And then they plate it again, and they consider whether or not it's resistant to particular types of antibiotics. So this classical paradigm, which I call plate and see, takes a long time. I mean, even though bacteria under ideal conditions, you might be able to get them to replicate every 20 minutes, say for E. coli, often it takes some time to go from sputum to colonies and then colonies to selective plates. And it can take weeks. If you were trying to grow mycobacterium, which can be involved in all sorts of diseases, including tuberculosis, it can take 2, 3, 4, 5, 6 weeks in order to figure out if you have the disease, right, the infective agent for that disease. And you also get what you plate for. So what you may plate out may not at all reflect the phenotype of what you might find in a lung environment or a blood environment or a urine environment. But that is the classical paradigm, and it is the gold standard at the moment, and I think we can do a lot better. Here's the new paradigm that I think we can reach. No sample preparation. Fast, we can figure out what we have on the order of minutes, and we can potentially have the phenotype reflected. Is it antibiotic resistant or not? And can we tell that in a matter of minutes? And the way we can do that is use Fenn's, error, I guess I wouldn't call it an error, but his contamination by smelling our culture using what is termed now secondary electrospray ionization, mass spectrometry, or SESI-MS. There is another name for this that's used by another group called extractive electrospray ionization, if you're curious. And so what we do with that technology is, instead of putting our sample into the liquid phase, we instead pass our volatiles, whether it's the headspace or breath of a sample, through the droplets once they've been formed in the electrospray. They will dissolve into the droplet, have a protonation reaction occur, and then they'll be attracted into the mass spectrometer, and then we'll have some spectral data, which I'll show you in a moment. So how do you go from, I don't know, a really grand idea into practice? This is what we've been trying to do in my group for the past 3 years. You do a whole lot of experiments in the flask to characterize the volatiles produced by your bacteria under different sorts of conditions. Then you do some work in mice. Once you're sure what you know you're looking for, and I'm very careful about using animals in experiments because I don't see them as just disposables. But once you know what you're looking for, you have to go into a system where you can infect a mouse and look for the signal that you expect for a particular type of bacteria and know that against a biological background. And you can imagine doing that on multiple levels, not just with one bug. And then you also do the same thing for humans, except you can't really infect them. That wouldn't be legal or appropriate. But you can look retrospectively at someone's breath or the sputum that they're producing and see, based on the clinical micro results, whether or not you've seen what you hope to see in terms of the bug volatiles. So I think this is a nice melding of a medical approach and an engineering approach. And what does that really mean? To me, it means that I get to use really cool technology. I have great instruments in my lab, mass spectrometers. They're big, they're expensive, they're impressive looking. There's some value in that. We identify things and we quantify them, and we do so in a robust manner. We can take some of our mathematics that we're not afraid to use in engineering and do model-based predictions. And then we have real applications for this. We're going for human health applications, among others. Haven't shown you any of the data yet, but I will get there. It's a big lead up. So these are the sorts of questions that we ask in my group. Who or what am I? How many am I? What are my biomarkers? And what is my metabolic status? Am I active? Am I latent? And for TB in particular, that's important. Am I responding to my host in some way? Is there an inflammation response, for example, that's measurable? Am I responding to antibiotics? Obviously an important thing to know. Am I undergoing evolutionary changes? I mean, we hope that we don't have bacteria that long in us that there are evolutionary changes, but in some cases, that's absolutely what happens. And maybe these volatiles are actually useful in other ways. Maybe they're antagonistic or synergistic for bacteria that are living their lives. All right, so we started off, of course, by doing flask experiments, and many of them. And we're just going to show you a subset of the data so you can get a feel for what that's like. Let's say we grew these, I haven't listed them here, but let's say we grow some bacteria, maybe in tryptic soy broth, a very common complex media for bacteria, rich media. And let's say we grow them for 24 hours and we sample the headspace of those bacteria. And we're just looking to see, can we tell the difference between one or the other? And so what you'll see here is a mass-to-charge ratio, which in regular people terms is just the atomic mass or the atomic weight of the molecule plus or minus 1 if it's protonated or deprotonated. And then on the y-axis, you have the relative intensity of the signal. So the signal that's at 100% is the one that's produced the most, and everything else is made relative to that, all the other signals. So we've taken some of perhaps the bacteria that you've heard about before, Pseudomonas aeruginosa. It's a common bacteria. We'll come back to that one later. Staph aureus, I think most of you have heard of. E. coli, of course, and Salmonella. And you can just look without knowing anything about electrospray ionization or even about these bacteria, and you can see there is a difference in terms of the suite of volatiles that's produced for each of these bugs. Some of them will have some of the same volatiles. Some of them will be unique. Sometimes the ratio between peaks is unique for a particular bug. But this is the sort of data, some of the types of data that we can get. We can also actually, because we have a triple quad instrument, take this peak, for example, blast the hell out of it using the third quadrupole and come up with a fragmentation pattern that we can then use to do identification. Because this is a new technology, soft ionization technology for small molecules, we don't have access to a major database in order to know for sure if we have what we think we have based on fragmentation analysis. So in order to confirm the identity of a compound, we have to do what people did in the old days, which is run standards after making predictions of what we think things might be, and then and see if our fragmentation patterns match, etc. So we did that. And here's from our very first paper using this technology. This is the Pseudomonas, the Staph, the E. coli, and the Salmonella. And this is just an indication of quantity that we find in the headspace after 24 hours of growth. And these are the compounds that we were able to identify. Some of them were known before, some of them are new compounds. But we were able to do that. It took us a long time to do this, and I'll come back to some technologies that we've been employing in the past year or so to make that discovery process faster. But nonetheless, we did an enormous amount, and are still doing an enormous amount, looking at different types of bacteria grown under aerobic, anaerobic, exponential phase of growth, stationary phase of growth, different nutrient conditions, and so on, in order to come up with fingerprint patterns like this, as well as molecule IDs. And now I'll tell you that for those 13 compounds faded out there, it took us a year and a half to really know for sure that these were what we thought they were. We certainly knew the bacteria were producing these peaks. We certainly knew that those peaks were for individual compounds, but identifying them took a long time. So naturally, we turned to even cooler technology, or as cooler technology, in order to identify those compounds more rapidly. So I'm going to show you some data from a technology that many of you will be familiar with, gas chromatography mass spectrometry. So we separate on a chromatography column, and then after that separation, the compounds are sent into a mass spectrometer for fragmentation. So we have retention time in the chromatography column here, and then relative intensity of the peak for particular compounds. This is one-dimensional gas chromatography. And this is an example of data from the headspace collected onto some fibers that are able to absorb the volatile molecules. And here's what you get. And you're able to, in some cases, identify what these peaks are. But in many cases, you have multiple things eluting at the same time with 1D GC. That can be a real problem when you want to identify the suite, the metabolome, if you will. So, and this is pretty cool, a lot of colors, so let me lead you through it. If you add in another dimension, as you might imagine, things start to separate incredibly beautifully. So if you imagine separating firstly with boiling point, and then maybe secondly with hydrophobicity, then what you get is a whole bunch of things that may be present in this peak that start to separate out in the second dimension. And I'll get to a specific example in a moment, but I want to show you some pretty color. What we can do then is all sorts of things with this data. We can identify biomarkers. We can look comprehensively at things that we couldn't necessarily see with the other method. And this is just a plot of this data shown a little bit differently. This is the first dimension, the retention time in the first column, and this is the second column. And what we're doing is we're tracking classes of molecules, in this case in the headspace of Pseudomonas aeruginosa, and looking at the variety of molecules that are present. Under 30 compounds were known to be in the volatile phase produced by Pseudomonas before we started our work. We have since identified hundreds of compounds that are present that may have all sorts of implications. Here's another look at that 2D data. I just think this is a cool way of looking at it to give you a sense that we can be quantitative as well. So again, this is a one dimension of separation. Here's the second. What you're seeing here is column bleed. I thought I'd show you some of the real data. Sometimes you have things that are still messy even with this system. How many people here have worked with Pseudomonas at all? Yes, yay. So Pseudomonas aeruginosa has a characteristic smell, right? Yes, and it's not a bad smell, is it? What does it smell like? Do you guys know? Well, I know you know, Carl, don't you? Grapes. Grapes. You wouldn't have guessed it necessarily. Some bacteria smell disgusting. But Pseudomonas, if you waft it, don't snort, but if you waft a Pseudomonas culture, which probably you shouldn't do, and I'm not advocating, then you will smell a grape-like odor. That grape-like odor is considered fairly characteristic for Pseudomonas aeruginosa, quite characteristic to the point of people thinking about using it to identify that organism in infectious disease. It's called 2-aminoacetophenone. And unfortunately, when you look at this in one-dimensional gas chromatography, it co-elutes with 8 other peaks. In fact, I've listed them here. So in one dimension, they're all going to be in one peak, but once you separate in the second dimension, they all fall out quite beautifully. And it's not produced in a very high abundance, but we're able to smell it. So 8 peaks are co-eluding in one dimension. So this 2-dimensional gas chromatography, it's a time-of-flight system, mass spec system, is very comprehensive. And here's the cool thing. It's database friendly. So each one of those dots can be blasted, not blasting in the biological sense, but blasting meaning break it apart, and fragmented, and then compared to international databases where you can be fairly sure that you know what you have, and then you could buy the right standard and test to see if you have what you think you have. So we now use these 2 technologies. The secondary electrospray ionization mass spectrometry. I haven't told you how quick it is. I will tell you because it's awesomely fast. And then we use a much slower technology to be really comprehensive. And what we do is, what we do in the flask or in the breath here, we'll do also using 2D-GC-MS. And we'll use the information here to tell us what these peaks are, so that we don't have to spend 1.5 years finding 13. And as I mentioned before, we have identified hundreds of new compounds. And it's not all bad news. You might think hundreds is overwhelming. All of those, some of them are unique. Some of them you find with other bacteria. But as a suite taken together, it's very, very useful for diagnostics. So how fast is CESI-MS? It is damn fast. We are not separating. We are literally sending the gas through the electrospray cloud, and it goes, once it's charged, goes straight in the mass spectrometer, and that's a very fast process. We haven't optimized this at all. We usually collect information over a minute. I think we'll probably get it down to 10, 20 seconds in the future. So we can take a breath sample or an off-gassing of a bacterial culture in less than a minute. So you won't have to wait 24 hours or a week or 5 weeks, ultimately, to do diagnosis. You will only have to wait minutes. And once we get the software integrated, maybe tens of seconds. All right, so our goal, as I mentioned, is infectious disease diagnostics via volatile analysis. That promise has been around for a while, but I think now we have the microbiological knowledge plus the analytical skills to really tackle this, this grand goal and do it, which is cool. All right, so as I mentioned, this goal requires that we work in the flask, in mouse models, and also in getting human samples as well. And I have 2 clinical stories to tell you about our progress at that level. A Staph aureus story and a Pseudomonas story, and I have to tell you that they're both related to each other. And remember that our questions are things like this. We just want to know who they are. Identify your cells by your volatiles. How many there might be, and then their biomarkers, and then the sort of secondary other grand goals are things about their metabolic status. Presently, the competing technologies, most of which are mass spectrometry technologies, don't have anything to do with metabolic status. So you might use a moldy technique where you would take a bacterial colony and you would blast it and look at the ribosomal RNA to do identification. But you'll never know, if you're doing a real sample, if that bacteria is alive or dead. And it really matters. Sometimes it matters to know if it's dead because it may have squirted out toxins, but you also want to know if it's alive, and squirting out toxins or other things. So I think we have an advantage in many areas because we can tell something about its metabolic status. All right, if you didn't know it already, you are covered with bacteria inside and outside. In fact, don't let this freak you out too much, you have 10 times more bacteria on you and in you than you have your own cells. So it's okay. Consider yourself a meta being, that's how I like to look at it, and that most of the bacteria are our friends. A third of us, if we looked at our skin or in our nose, would have Staph aureus, particularly common up here. And Pseudomonas is everywhere. Presumably, many of you have taken a shower in the last day or two. And in that shower, well, maybe you're working hard on something and haven't had a chance, and in which case you will have different volatiles. But if you took a shower this morning, you would have bathed in Pseudomonas. So it's not too bad. If you're immunocompromised, maybe you're undergoing cancer treatment, for example, then Pseudomonas aeruginosa may be opportunistic, and it may come in and take residence in your lungs, for example, or in a wound. Let's talk a little bit more about Staph aureus. So MRSA, most people have heard of. That's the thing you don't want. You get a MRSA infection. You can look it up on the internet. I didn't bother to bring a slide with the disgusting pictures of MRSA infections. They're vile. They're difficult to treat because the bug is fairly antibiotic resistant. About 500,000 people get an infection a year, and it's very expensive to treat something that's methicillin resistant. Acute infections with Pseudomonas are common if you have a cut, for example. And chronic infections, which can occur in patients with cystic fibrosis or chronic obstructive pulmonary disorder, are very challenging to understand and to treat. And of course, they're also expensive and cost lives and cause great illness. So how about this? Pseudomonas and Staph are the dominant gram-negative and gram-positive causative infectious agents for microbial disease, just hands down for humans. TB is a big one too. I'm thinking mostly in terms of the West at the moment. So we ask a very simple question in the beginning. Can we tell the difference between Pseudomonas and Staph based on the volatile metabolome? And of course, we first ask that question in the flask, and I've already shown you this data, and it's true for all sorts of different media, although some of the peaks will come and go. Some of them are core peaks and remain. And again, we're looking at mass to charge ratio and relative intensity. And again, without knowing anything about mass spectrometry, the patterns are different. And as I said, this holds true for different media, different conditions, and so on. All right, fine, it works in the flask, and you've done a lot of that testing. So what about in the mouse? So we've taken, for example, here this data, black 6 mouse. We've infected the mouse such that it will get a lung infection, and we've monitored that infection at the 24-hour point. So it has quite an infection at that point, but it's an acute infection with Pseudomonas aeruginosa strain PA01, which is a classic strain that's used in labs that reflects an acute infection isolate. A methicillin-sensitive Staph aureus, which is reflective of a lot of Staph aureus infections, and then an uninfected mouse control. And again, we have the mass to charge ratio, or the atomic mass, basically, of the peak, and the relative intensity using the rapid profiling SESI method. And again, you can see that there's a difference between, this is interesting in and of itself. This is a mouse. These are all mice on ventilators after the infection has taken place. And there's a lot of information here about inflammation that I don't know much about yet, so I can't speak to it very well, but I thought I'd throw it up there. We're talking about 5 mice replicated here, and all of these are very reproducible. And there's a difference between the two, as you might expect based on the flask data, but it's very nice to see this in a complex background. And I'll show you some comparisons between flask and mouse data in a moment. All right, so I came at this a little bit sideways through learning about cystic fibrosis, because at the University of Vermont and also here at the medical school, there's an emphasis on learning about this particular disease and learning how to treat it, learning how to diagnose infections, and understanding how infections contribute to the morbidity and mortality. So we asked this question, and it's a far more complicated question, because what we're asking is, can we tell the difference between an acute and chronic infection strain of a bug? So I've told you or shown you data about genus level. What about strain level? Things that are quite genetically similar. Things that metabolically, therefore, will be quite similar, and yet not exactly the same, because a chronic infection strain has typically lived for a long time in an environment and evolved. Here is an ugly picture. If you don't like to see nasty pictures, close your eyes. If you're too curious, sorry. Ah, that is vile, but it's real. This is the lung of a patient who expired, obviously, who had cystic fibrosis. These folks don't get to live a very long time. The average lifespan is up to about 40 years, which is far too close for my comfort zone. And what you see is bronchiectasis, which is here, or as I say, bronchiectasis, which is just flabbiness associated with dealing with infection. You see mucosal plugs, and you see hemolysis due to bacterial infection and fighting bacteria. It's a war zone in that lung. And this is a lung of someone who lived and tried with difficulty, obviously, to breathe. And frankly, knowing whether the bacteria are producing mucus plugs or not is important in the treatment of cystic fibrosis patients and COPD patients as well. So I'll remind you that in order to figure out what bacteria are present in patients who have chronic infection, you have to cough up stuff, plate it, plate it, plate it. But let's look at some interesting, even though it's in a human and you've seen what it actually looks like, some interesting microbiology. This is a patient whose sputum was tested for the presence of Pseudomonas from the age of 6 months up to 96 months. 6 months. They were infected, colonized with Pseudomonas aeruginosa, and 90 months later, we were still measuring the Pseudomonas, not me, it's a royal we. We're measuring the strains of Pseudomonas. This is a phylogenetic tree tracking the mutations that have taken place over time as the bacteria has evolved to evade the lung immune system response. So there's evolution going on for patients who have a chronic infection of the bacteria that are present. And I'll tell you now, there's more than just Pseudomonas present, but nonetheless, Pseudomonas is considered one of the big, probably the biggest bug in the CF world, and COPD world. So there are mutations that have risen and gone away, but over time, there were 35 mutations that were acquired by the bug living in the lung of this patient. And that seems to hold true for many patients, whether they start to be infected at 6 months or later. What are some of these mutations? You can imagine what they might be. Greater resistance to antibiotics, for example. And in this case, one of the mutations that we focus on in my group is a transition from colonies that look like this to that nasty looking slimy stuff. So this is a transition to mucoidy. And as you might imagine, treating this is a lot easier than treating bacteria that are hiding in the slime and who are probably somewhat microaerophilic, maybe even aerobic, and there's all sorts of redox stuff going in there, making antibiotics less effective, and so on. And indeed, what you find is that most patients at some point will transition from mucoid to non-mucoid, non-mucoid to mucoid in the lung. So of course, we wanted to look at that very complex question and see if we could make some insight. So we grew a bug that is reflective of this phenotype. That's the PA01 strain that I mentioned before. And then we took a cystic fibrosis isolate that we know is indicative of a chronic infection and produces a slime called FRD1. And we grew them in synthetic CF, cystic fibrosis sputum media, which just contains a lot of amino acids, which is what you find in the lung environment after the war has taken place and during the war. And what we find is that the one reflective of acute infection doesn't really enjoy being in that environment terribly much in terms of producing volatiles. But of course, the one that has evolved to live in that system is having a party. It's enjoying all the amino acids. It's having appetizers, the main course, and dessert. And we found this to be true for many isolates that are from chronic infection versus those in acute infection. So of course, that's great in the flask. But what about in the lung, in this case of a mouse? So we've taken the same mouse, a black 6 mouse, infected it for 24 hours, in this case with strains of PA1 and FRD1, 5 mice apiece. And we vent the mice, so we're collecting the breath of the mice and then expunging that breath into our secondary electrospray system and looking at the peaks. So mass to charge ratio, relative intensity here. And what do you know? They look fairly similar. But if you look down amongst the bushes here of the spectra, there are actually some significant differences between the chronic infecting strain and the acute infecting strain. And we think that these bushes are actually going to be some point, we're not there yet, able to be used to tell the difference between whether you've acquired a particular mutation or not. And I'll show more detail on that in a moment. But what's awesomely cool is that against a biological background, we can tell the difference between strains. Okay, I never thought, hoped maybe in front of my students said yes, but I really wasn't sure if we'd be able to tell the difference between bacteria at the strain level using their volatiles. We've been able to show this for Salmonella, for E. coli cerevars. Okay, so that's just antigenic differences. It's totally cool, very exciting. All right, so now let's look at just a quick analysis of the mouse data now with the Staph aureus strain, the acute infecting strain, and the chronic infecting strain. Some of the peaks are in common, and we're very conservative with that cutoff here. So this is very real data. There are some peaks that are unique to the acute infecting strain, some unique to Staph. Staph is actually a lot more, I've got a growing appreciation for Staph. It produces a lot of really interesting volatiles, which allows us to then separate it from other bacteria much more easily. And then FRD1, which has evolved to live in the lung environment. So can we distinguish between acute and chronic infection strains? In the flask, yes. In the mouse with data just from 2 strains at this point, yes. But it's going to require a lot more work than just what I've shown you. So I have an incredibly talented postdoc, Dr. Heather Bean, who has been, she's actually a chemist and a microbiologist, but using a very systematic sort of engineering approach to start to answer the question about what do we need to look for. What are going to be the biomarkers for those particular mutations that we see in CF and COPD? And so she's been working with a more virulent strain of Pseudomonas that people often use for CF work and others. And she's been doing some deletion mutant work. So she will knock out a gene that's associated with the mutation that we see in CF through chronic infection. And then we've done a lot of 2D-GC analysis of that, as well as the SESI analysis. Her ambitious project is to map the biomarkers to the mutations. And I'm pleased to say that just a couple of days ago, she was awarded a very prestigious fellowship from the CF Foundation to do that for the next 2 years. So she'll work with single deletions. She'll compare them to each other. She'll work with double deletions and then mixtures of mutants together, grown together. It's a very ambitious project. If she can do half of it, it'll be freaking amazing. Okay. And she is amazing, so she'll probably do it. How about this question, which is also a very refined question, maybe even more difficult than asking the difference between strains. I mean, a chronic infecting strain, I know it's already got at least 35 mutations, so I would expect there to be some differences in the volatiles. MRSA, the methicillin resistant versus the methicillin sensitive. Well, I did this to see if we could see a difference, because I didn't think we would. So the difference between isogenic sets of these is a MEK-A cassette. When you look at the MEK-A cassette genetically, you wouldn't think it would have any real influence on metabolically what's going on in the bug. Well, whatever the heck is going on, actually I'm Australian, I say whatever the hell, but whatever is going on, it's reflected in the volatiles. And so we need to work with folks who really know Staph in order to ask fundamental questions about what may be causing the differences. But here are isogenic sets, so they're genetically identical except for the inclusion of a MEK-A cassette. And what we see in the flask, I believe this is in tryptic soy broth again, after 16 hours of growth, is that, well, they look similar. There are some peaks that look the same. It's pretty hard to tell, I think, just looking at the spectra here. But you could probably, by eye, because the human eye is wonderful at pattern recognition, see that there are possibly some differences. And these are all normalized by cell count and things like that. Fair enough. I'll show you presence-absence analysis in a moment. Let's look at the same strain grown in the flask compared to in the breath of mice. Well, my word, some of these things are in greater abundance, and some of them are unique. What does that mean? If we look at the peaks that are present in the breath of a mouse versus in a flask, we find that 36 of those peaks are in common. Maybe they become the fundamental fingerprint for this bug, and we've done a lot more tests to underscore that there are about 30 compounds that seem to hold true no matter how you grow Staph. 12 appear to be host-related, in terms of the infection and the host, and 28 are associated with just growth in tryptic soy broth. So some of our next steps, and some of these we've already done, but some are certainly associated with things that are ongoing, are certainly looking at an even greater variety than aerobic stationary phase. I didn't write down exponential phase and anaerobic growth for these organisms, because you might imagine in a lung environment, for example, if you're in slime, then you're going to look differently in terms of volatiles than if you're the outer edge versus the inner edge. So we need to keep building our database just in the flask. And looking at mixed cultures. So most infections are not a monoculture, although they typically are dominated for acute infections. So you might have a Pseudomonas dominant culture or a Staph dominant culture. Maybe a few other things are trying to get in there as well. For chronic infections, it's a completely different story. There's usually many, many microbes, and we have no idea about their interactions quite yet. But these are some of the bugs that we work with in my group. Our favorites are here, although I have to admit that Mycobacterium is becoming one of my favorites. It's such an interesting organism. But Strep pneumoniae, Legionella, Haemophilus, Klebsiella, for example. And we don't grow influenza in the flask, obviously. But I'll come back to that in a moment. So let me show you some of the co-culture work that we have done. So we asked this question. If we grow Pseudomonas and Staph aureus together, will we be able to extricate out of that co-culture volatile metabolome the signature for Pseudomonas and the signature for Staph? And can we use that information not only for identification, but also to quantify what's present using the rapid profiling technology? So this is a portion of a principal component analysis. The first principal component is here and the second is here. I'm sorry that I blocked out the percentage. But what we've done experimentally is we've looked at different amounts of bacteria at the final endpoint that are the composition in the flask. So 100% Staph is reflected here in the principal component, 100% Pseudomonas aeruginosa. And then we can see that there is a reasonable linear relationship. And if you want to look at particular compounds, you can get a really high R squared. If you look at the suite of compounds as a cohort, then your R squared is still pretty damn good, above 0.85. So we can actually grow them together and take the volatile metabolome of those two cultures grown together and then look at what we might have predicted based on the proportion of cells that we find at the end. So we've grown them, then we plate them out and we say there are this many Pseudomonas, this many Staph. And then we use the initial spectra that we get on single cultures to predict what proportion we should see in our final spectra. And we were able to do that pretty well. We of course found that when grown together, that some unique and interesting volatiles are produced, and we have no idea what that means. But it's a very interesting point, and if I come here and you want to work on a PhD, that would be a really interesting project. And as I mentioned, we can quantify. So I've shown you just a little sampling of data indicating that we can separate bacteria in the flask and in some early experiments with mice down to the strain and cereval level. Strain in the mouse with Pseudomonas and cereval level in the flask for E. coli, Salmonella, and some other bugs, but not in a mouse background. We've done mixtures as well as co-cultures. So if you take a Pseudomonas culture and a Staph culture, and you know how many of each you have, and then you mix them together, which may occur when you have volatiles in the lung. If you have a Pseudomonas infection here and a Staph infection here, you may have that situation rather than a co-culture. So I've shown for mixtures and co-cultures that we can quantify and pull out fingerprints. We've looked at mouse breath. I can't yet show you the sputum and human breath data. There's a little bit of intellectual property stuff associated with that. But we have looked at human sputum and found our signatures of Pseudomonas in human sputum. And we think we can see Staph as well, though we don't have enough data to really statistically prove that yet. And we've just started work with infectious human breath. So we've done some other work in the past looking at human breath through starving yourself for a little while, looking for acetone peaks and these things that people already know how to do in order to know how to sample human breath. And now we're looking at breath with infection. So where am I going my group rather. I just write the proposals and they go in and do most of this. We have some questions that are far more clinically oriented that we need to ask in the mouse as well as the human. One of the big questions clinicians have is it a viral infection or a bacterial infection? So we now feel that we're capable of answering that question in a mouse model. And it's really important, do you give them antibiotics or not? And certainly, if it is bacterial, then we are figuring out what the signatures are so that spectra, as well as the biomarkers, and not just saying we have a peak, and if you see these peaks, then we know that you have a particular infection, but also identifying what those peaks are. For the major lung pathogens, a lot of people work on just one bug, and that's cool, but in infection you have to look at many, and that's what we need to do, and it's really fun for me because I like looking at lots of bugs and learning about them. We're also monitoring for clearance, so we want to know and ultimately be able to look at inflammation. But we want to know if we can track an infection that's cleared, either naturally or through antibiotic dosing. And what we expect is that you'll see the rise and fall, for example, of Pseudomonas fingerprints. And then you'll see concurrently, but maybe a little delayed, the inflammation response reflected in the volatiles. I'm not an inflammation person. I look at things through bacterial eyes. Doesn't really make sense, but you know what I mean. But I have some recent collaborations where people do know about inflammation and will help us understand whether we can use that information not only to track the influence of infection on lung health, but even something as simple as, is somebody being damaged by being on a ventilator? Something as simple as that without any microbes, although people on vents get all sorts of infections as well. So these are the bugs we're looking at, including influenza. And we're going to be starting that work in early April. And of course, on the human side, we now have approval to work on breath, and we've been looking at sputum for some time. And our target is to go after the big two, Pseudomonas infections as well as staph infections. And my word, if we can just tackle those two, it'll be very cool. So again, these are the sorts of questions we ask in my group. And we've pretty much been working in this area. Those are the questions you have to be able to answer first, who they are and all that kind of thing, how many. And we're just starting to learn how to ask questions in this area. So these are some more fundamental research questions that are very clinically relevant at some point. As I mentioned, for TB, you want to know if the infection's latent or it's active and you've got a patient that's running around potentially infecting people. Requires money to do this work, and I've been really fortunate to have funding from the CF Foundation, the center on campus that's in immunology and infectious diseases, and also NASA. That's pretty cool. I wanted to mention very briefly some of the other volatile projects we have going on, because we're not just focused on human health, although that's predominantly what we do. So I thought I would list some of them. There's a lot of dairy in Vermont. People care about the health of their dairy herds for good reason, because they care about their cows and the milk production. So I have a project with a veterinarian looking at mastitis detection, just from looking at milk infected, if you will, with Staph aureus in particular. We've done a huge amount of work in the food safety area, where we've looked at trying to identify E. coli ceravars, like O157H7, against a background of Salmonella, Staphylococcus, and soon Listeria and others. Ventilator-associated pneumonia is a very big deal. So being able to, if you get put on a vent, don't ever get put on a vent. But if you do, chances are you'll get a staph or a Pseudomonas infection. And knowing when you have it and being able to treat it quickly is incredibly important, because you're already very compromised at that point. I'm working with a company to commercialize some of this stuff in the clinical lab. And then I've just started to work with people on some of those fundamental questions. I've focused a lot on the technology development and knowing how to ask clinically oriented questions. And so I'm now partnering up with really cool people to look at some very specifically focused questions in their areas of expertise. So knowing if E. coli has a particular type of lipopolysaccharide, so on its outer surface, can be important for clinical diagnostics, linking microarray data with the metabolome, something I don't know how to do yet, but working with someone to do. Skin stuff. Who knew people were interested in the bugs on skin? But there's a whole field of people interested in that for odor control, among other things. I don't know much about it. This one's cool. So maggots are still used as therapy. I know it's gross, but they are. They're very effective at cleaning out wounds, particularly the dead stuff in them. I wouldn't want to look if it were me, but they're really good. But it turns out that they are repelled, I don't understand why, by Pseudomonas. How can anybody be repelled by that bacterium? And they're attracted to the other things like staph that you might find in a wound. But it turns out, if your maggot eats Proteus mirabilis, then it doesn't seem to mind going to the wound, which it's attracted to by the volatiles produced by the bacteria, and then can eat Pseudomonas. So we're working with folks to do that. And we're in Vermont, and we are looking at the age of cheese. I like cheese. I'd like to do wine in here too, frankly. Anyway, looking at whether we can tell the age of cheese based on its volatiles. This is an undergrad-driven project, a very cool one, where we look at 3-year, 2-year, and 1-year aged cheddar, and we can tell the difference between those, and we think we can tell the difference between whether it was fed on grass or silage. And I work with lots of people to do these projects, of course. These two folks at the University of Vermont. This chap here is at Spelman College in Georgia. This is Massachusetts General. This company's in Connecticut. This guy's at the University of Guelph, which for the longest time I thought was in the Netherlands. Turns out it's in Canada. This guy's in Australia. This is a French company. I suppose that makes sense. I don't know. This fellow's at Harvard, and this is local. But I get to generate fun ideas, and these are the people that do the work. I have a range of undergrads, master's students, PhD students, and postdocs, and they've all, contributed to making this project, or making these goals a reality. So with that, I will take any questions.
So you mentioned wanting to distinguish between viral infections and bacterial infections near the end.
Right.
And I'm curious, could you, using the volatile metabolic products, distinguish between a virus-infected host and a non-virus infected host?
That's the idea. I think it's going to be possible because the inflammation response is different. So, I mean, I've just started work to try and understand what it really means to be infected by a virus. Viruses are cool. Bacteria are really cool. Viruses may be cooler. I don't know. So I think in terms of immunological markers, there's absolutely a difference. How that translates to the volatile realm, I don't know. We've just started work with a collaborator in Sweden who measures aerosols. And at some point, we should be able to do aerosols in our system, but I don't know anything about them. So that combination of volatiles and aerosols will probably tell us enough. I'm cautiously optimistic. Good if we could.
Where is this in relationship to actual clinical application? What might it take to get there?
So the question was, where is this relative to clinical application? The first application I'll be trying to get commercial partners associated with. That presentation is actually in 2 weeks. And the company and I think that the best target is the clinical microlab. So it's not breath analysis or urinalysis or blood analysis or anything, sputum analysis yet. It's taking what usually takes 1, 2, or 3 days of culturing down to the hours level by doing high throughput screening of cultures growing on a proprietary media. So that may happen. I mean, I don't really know. It looks like FDA approval for this because it's similar to the end goals of what already exists in clinical microlabs. Is going to be a fast track, not the really slow process. And so maybe within 5 years, if we maintain our level of productivity and showing we can do what we want to do. I'll let you know in a few months if we get the funding from the company to do what's required, which is thousands and thousands of isolates. That's not something I want to do in my research lab, but through my affiliation with the company, that will be possible. I hope 5 years. The rest, maybe 10. For acute infections, chronic infections, bets are off.
Yeah, this lends some credence to animals that are used to detect diseases.
Definitely.
Also, it occurs to me you might detect diseases which are not yet presented.
Right. So I'm really excited about infectious disease, and it's very hard not to also be excited about other things. I get excited about a lot. So I've been approached by all sorts of folks for cancer diagnostics, metabolic tracking of disease. We're going to stick with infectious diseases of the lung as well as food safety related things and animal equivalents. But there's no reason why you couldn't potentially predict the onset of a disease by looking at things early on, and then also the amelioration of that disease. So one of the big issues in cystic fibrosis is when a patient has an exacerbation, which is usually microbially underpinned, they don't know how long to keep them on the multiple antibiotics that they aerosolize in and intravenously have them take. And knowing that endpoint, nobody's been able to figure it out at all. It's somewhat arbitrary and dependent upon the clinician. That's probably the bigger challenge. Maybe you could do it for vented patients or other things. But it won't do everything, this sort of technology, but something like it would be totally possible.
Is there anything about glucose that would allow you to tell from a volatile or a diabetic?
If I can put on a slight business cap, that market's fairly cornered with rapid technologies to detect, if you have acetone production or whatnot. But certainly we can find acetone signatures in the breath that indicates when somebody is the equivalent of diabetic. So we've just done it through having some of the students in my group fast for a period of time and then looking at what happens during fast and then post-fast, which is the same kind of idea for diabetics.
So one could do glucose testing without pricking their finger? Is that?
Yeah, which people would like, for sure. I think, so I'm not like a MEMS person, although I find that stuff really interesting. And I've just started to work with people who might be able to take some of these biomarkers and use them on little thin film devices, for example. As much as I would love to see everybody with a mass spec, the reality is miniaturizing them and getting them into the clinic may not be the right way to go anyway. But certainly the biomarkers that we find can be put on other platforms. And maybe that's what it would look like for diabetics or other things.
How do you collect mouse breath?
Very carefully. Well, it's an unfortunate thing for the mouse. But what we do is we give them a little tracheotomy, and they're then on a vent. And that allows us to collect the breath over some period of time. And we typically collect the breath in these Teflon-lined bags for anywhere from 30 to 50 minutes to get enough breath to do a lot of the analyses.
So how would it work for people?
Direct, well...
You would just breathe into a bag?
What
would you collect it? Where would I go like that? Or are you going to...
Oh, sure. Not am I going to continue? Mice are important, too. So with people... We've done work directly with the SESI system where you breathe right into the instrument. We just can't bring in, critically ill patients and expose them to all the people that we have in our school. Lots of gritty things. So you can breathe into a bag, take it over the instrument, squeeze it out, or you can breathe directly into the SESI instrument. Not true for the two-dimensional gas chromatography. That's different. But like I said, I think the grand goal would be, like I said, as much as I like the mass spec thing, probably it's just going to be the biomarkers put onto some device that you can breathe onto, and that's, like a dipstick test for urine.
So Mary, answering the question, how many am I? Is that just simply integrating area and the signal from the two-dimensional chromatography?
Basically.
So what about in vivo samples? Have you looked to see whether or not
Yep.
you can quantitate in vivo?
Yeah, but early days, and we have not done co-infection. We're just writing up a protocol to do co-infection, with staph and Pseudomonas, for example. But typically, close your ears if this is vital to you. We'll take the mouse lung, grind it up, and do plate counts, and then see how we've done. It's for the good of science.
No more questions. We just thank our speaker one more time.
Thanks.